Good evening. We're working on a quick revision for some of the important topics of Unit 4. We're gonna revise through questions. So I'll be solving questions about each one of those topics we took in Unit 4. Plus, at the end of the class, we will have a test. Just like what I told you last week, we're gonna have a test about immunity. Okay? So let's start with my first question. It's talking about photosynthesis, and it says here, explain why light energy is converted to ATP. That's like one of the first things we did in Unit 4. We stated that ATP is essential for all living organisms, including plants, because light energy itself can't be used by the plants for life processes. So plants do it like this. They take the light energy, they convert it to chemical energy, and that chemical energy is in the form of ATP. So I'm gonna write it like this. I'm gonna say ATP is source of energy. Light energy, on the other hand, it cannot be used directly. What does directly mean here? It means we're going to turn it to another form of energy, which is chemical. I mean here by chemical energy, I mean glucose. And then the glucose will be respired, turned to ATP. And that ATP here, just like in your body, use ATP for all life processes. Simply because it's way more useful. What makes ATP like a universal energy currency? Small, recyclable. It could be easily like passed from inside the cell to the outside and the other way around. So it can be easily transported. Now, for my case, for photosynthesis, you make ATP in the light-dependent reactions, and then you use that ATP in the light independent. So it's a product of light dependent, and then it's going to be used for the light independent. Do you guys know when exactly I use it for the light independent? I use it in the light independent the time when I'm converted GP to GALP. That particular step requires both NADPH and ATP. That's when we go and turn GP into GALP. Okay? Any question here? My second question, still in question one, they wanted me just to complete the equation for ATP, and they want me to name this reaction when we break down ATP. So it's ADP plus inorganic phosphate. This is called phosphorylation. You may call it photophosphorylation because we're talking here about plants. And the opposite reaction, what am I going to call it when ATP is broken down? Breaking down reaction, whenever you break down any molecule, in this subject, we call that hydrolysis. Okay? So you may also call that condensation, but because we're talking about ATP, we usually call it phosphorylation or photophosphorylation to be more accurate. Okay? So this was the first page. Now, second page. They asked me to explain the role of light energy in light-dependent reaction. While I was picking those questions, I was trying to find the more direct questions and the repetitive ones. So those are the ones that come more often. And you can get hold of those easy marks, secure those easy marks in every exam paper. So my case here is say, explain the role of light energy in the light dependent. Like light energy is needed to excite the electrons in chlorophyll. And it's also needed to split that water molecule, photolysis. So Both cases, that is essential because just what we said there, this is needed to make those valuable products reduce NADP and ATP. So one more time, I'll state that light is needed to excite electrons from chlorophyll. Light is needed for photophosphorylation. That's why they did not write photophosphorylation the key in the previous question, because it would be help you to answer the next question. And it's also needed for photolysis. And this is essential because those hydrogens you get from water, those hydrogen ions, protons, they are essential to make reduced NADP. I'm talking here about the products of light-dependent reaction, reduced NADP and ATP. Okay? Now, those are going to be used later on. Where do we use the ATP and reduced NADP? We use them together during the Calvin cycle, the light-independent reactions. Okay? My other question right here, it's like a multiple choice. They said the light-independent reaction used products from light reactions. Which row in the table shows you the product of light-dependent reactions? So, like with photosynthesis, we have two formats. We have the cyclic and non-cyclic. Just be aware of something, that when it's cyclic, the whole purpose of cyclic phosphorylation is to make ATP. Okay, so here's like a quick comparison between the two forms, light reactions. If it's cyclic, it says no NADPH is made. That goes with cyclic. So I'm going to take this and I'm going to put it here at cyclic. Okay. Where am I expected to make ATP? Now, this is controversial, but you make more ATP with the non-cyclic reason, simply because the chain is longer. You get more electron transport chain and you get more protons being built up there. Whereas no water is needed, that is cyclic. Why don't need water? Because the electrons are running in a cycle. So you don't need to replace any missing electron. Even there is no oxygens released here. Why again? Because there's no splitting of water, no photolysis. Which one is aiming to reduce carbon dioxide? Here. With non-cyclic, we have light reactions and light independent reactions. So we do make NADPH and we do reduce carbon dioxide to make sugar. But the cyclic, the whole purpose here is just to make ATP, nothing else. Okay? Now, going back to my question, what am I supposed to pick? I need NADP and I want to take them to the light independent reactions. Therefore, I need to pick reduce NADP and I need ATP. I need both products of the light dependent reactions. I can't choose cyclic because it doesn't make reduced NADP. Okay? Does that make sense? Now, this one, I think it's pretty simple. They gave us like the general formula of all carbohydrates, and they're asking me which inorganic molecule is needed to make carbon, hydrogen, and oxygen. Think about it. Where do you get the carbon from to make sugar? You get it from CO2, okay? Where you get the hydrogen from? You get it from water. And where you get the oxygen one, now this is also controversial because sometimes students think, well, oxygen comes from water. No, the oxygen in water is a waste product. Photolysis gives me oxygen, but that oxygen is not useful. It's going to be lost. The oxygen here comes again from carbon dioxide. And right here, they gave me these biological molecules and they want me to tell them, does that require nitrates? It does, right? Proteins got carbon, hydrogen, oxygen, nitrogen. So where you get the nitrogen from? You get it from nitrates. Okay. What about nucleic acids? Nucleic acids got phosphate groups and got nitrogen in it. So I'm gonna pick this. Triglyceride, they did not say phospholipids. Triglyceride. Triglyceride is just carbon, hydrogen, oxygen, just, right? So for that reason, I'm saying neither. This will only relies on carbon dioxide and water, nothing else. You don't need any component from the soil. And this means we're done with the first question. Do you have any doubt about what we've done so far? Second question is this one. Still photosynthesis. It says explain how light-dependent reactions enable hydrogen to be incorporated into gulp. Do you know what they mean by getting incorporated? Meaning where you get the hydrogen to reduce GP and form gulp. As if someone's telling you like this. So again, that is light reactions here, light-dependent reactions. Light-dependent reactions give me this, give me ATP, both of which are needed for that particular step. The step where I reduce GP to gulp. So I'm going to state that one here. I'm going to say the hydrogen that goes into gulp comes from the water. It's like the previous question. Water is split during photolysis. This gives me two things, two valuable things. One is the electrons, and second are protons. So you get protons and electrons. Both are valuable. Now, this question is asking me about the proton part because the question says hydrogen, okay? So electrons gonna return back. So the electrons in the light reactions are gonna be used to replace. Do you know when you excite chlorophyll, you're gonna lose an electron, right? You excite it, you lose electron, but the water is gonna replace that missing electrons. Okay, I'll write it here. So the electrons replace those ones lost by chlorophyll. So here we're gonna make reduce NADP. And as I just stated, that reduced NADP is going to be valuable for what? It's going to be valuable to convert GP into gulp. Okay. The bottom question is asking me about the factor of temperature. Now, with photosynthesis, every limiting factor has something to do with the light reactions or the light independent reactions. So, for instance, someone asked me about temperature. I will talk about the light independent reactions. While with temperature, I talk about Calvin cycle. That's because the enzymes for the Calvin cycle are floating in the stroma of the chloroplast. Those enzymes are quite sensitive to changes of temperature. Think of the only enzyme we have in the syllabus related to this. Think about Rubisco. Rubisco is gonna be affected by temperature. So changes of temperature affect light independent. Carbon dioxide still light independent, but obviously when it comes to light, that is for light dependent reactions. So going back to my question, they're asking me about temperature here, right? So I'll talk light independent reactions. So in general, someone asks me about temperature. Since your early years, if someone asks you about temperature, you tell them there is more kinetic energy for enzymes and light independent. And the only enzyme I have to be stated here is Rubisco. Therefore, again, I'm talking enzymes here, right? So what are those key words we always say here? We say collisions. And think about it now. Collisions between what and what if I'm talking about Rubisco? Rubisco reactions like this. It's CO2 combining with RUBP, okay? This particular reaction. So those two are the substrates of Rubisco, okay? So more what? Enzyme substrate. Together, the enzyme substrate will form a complex, okay? So more GP turning to what? To gulp again. Obviously, I'm writing like many extra points in the exam, as we always say, the number of marks plus one. You write 4 points if it comes 3 marks like this. And I'm getting to the other part here. This one, let's take a look at the graph. It's rate of gulp production. In other words, the rate of photosynthesis, the rate of light independent reactions. It's been done twice. One with plenty of light and one with limited amount of light. And what are we measuring? We're measuring the rate of uptake of CO2. Did you notice that I have negative values for the uptake of CO2? What does it mean if you have negative value of uptake? So if uptake of carbon dioxide is in the negative side, that means that the plant is not uptaking CO2, the plant is releasing CO2. So sometime here on that graph, I got negative uptake. That is time when the plant is actually releasing carbon dioxide. Think about it. When is the time that the plant will release CO2 rather than uptaking? That's when the rate of photosynthesis is slow or not occurring. What process is responsible for this? The process responsible for the release of CO2 is going to be respiration, aerobic respiration. Anyways, so the question is asking me here, explain why plants grown in the light had a greater uptake of CO2 than those in the shade. Pretty obvious. You're going to talk here about what? Talk about light reactions and how are the light reactions supply the Calvin cycle with reduced NADP and ATP. And it's saying here above 26, meaning they wanted you to talk about this part here, start from here. Okay? So I'm going to say it like this. Plant grown in the light have more light energy for the light dependent. Light is needed to excite chlorophyll and photolysis. So more two products, ATP, reduced NADP, okay, which are to make they, which are need to supply with the light independent reactions. Now, on the next page, they're going to ask me about that negative carbon dioxide uptake. So, going to have to talk about respiration. So let's get there. So they're saying here, like, suggest why the net uptake of carbon dioxide was negative at the beginning. Again, plants are like this, right? If there is limited supply of light, or it's in the dark, then the plant will be releasing carbon dioxide rather than uptaking carbon dioxide. I'm going to say here, because respiration is at a greater rate than photosynthesis, RUBP is accumulating. I have plenty of RUBP and limited supply of carbon dioxide. So that carbon dioxide here is building up. Again, it's a cycle. If the RUBP, and I have here that CO2 to give me the GP. Now let's assume something. Let's assume that GP is not turned into gulp. Okay? Why is not turned into gulp? Because I don't have enough of those two. Okay? I don't have enough ATP and NADPH. This is accumulating. Carbon dioxide doesn't have anything to bind to, right? This is building up. So there'll be no further uptake of CO2. Okay? So this means we're done with the second question. Question 3 talks about succession. It starts with a definition. They're asking me to define climax community. What is climax community? Climax community is the point where you reach an equilibrium, meaning stability. The ecosystem is stable. I have the same number of species. I have the interactions, and I have a dominant species. Usually the large trees form in the climax community, and I have plenty of diversity. So I'm going to write the definition here. I'm going to say it is the final stage of succession, and it is when the community becomes at an equilibrium or stable. Next, they're asking me about the stages of succession. So I have those stages in front of me. They said using information in the diagrams, explain the changes from stage 1 till stage 3. As if someone is asking you, write what happens to the soil from the time when succession starts, excuse me, from the time succession starts with pioneer species until we reach the climax. So I'll first start like this. I'll tell them the habitat will be first inhabited by what we call them the pioneer species. Give examples. What are examples of pioneer species? Could be lichens or algae. These are known to be quite resistant to abiotic factor. They can grow with minimum amount of organic materials. So they will grow some roots. They'll penetrate the soil. Sorry, they'll penetrate the rocks. There's no soil yet. And they'll start to make the soil. What happens after the pioneer? Right after the pioneer, we're gonna get the small plants. Those small plants will soon decompose and they're gonna make the soil thicker. We're now getting organic nutrients in the soil. Then comes the larger plant. And again, when these die, they're gonna decompose and make the soil even thicker. Finally, we get the large trees at the last stage, the climax. Now they're asking me this. Now I have that pioneer species, okay? What effect will have if I got some herbivore into the this stage of the succession? So again, at the early stage, somebody has introduced herbivores into the early stages of succession. How is this going to affect the progress of succession? Now, obviously, the herbivore is gonna feed on plants, including the pioneer species. So if it feeds on plants of the pioneer species, they're gonna slow down the process. I'm gonna get less soil. Even it's gonna feed on the leaves from the larger trees. So all of this will slow down the progress of succession. We're gonna get less soil. We're gonna get less plants to enrich the soil. So I'll say the herbivore is going to start to eat those pioneer species. So I get fewer plants. Therefore, the soil will not be improved with nutrients. The herbivores would also feed on the shoots of the larger trees. For that reason, you would expect the climax to take longer. And we're done with question 3. My fourth question is taken from the topic of ecology. It talks about an animal. Looks, they call it the Tasmanian devil because of its looks. Look how it looks like. Anyways, so they said that this guy here is suffering of cancer. They're saying it has some fatal cancer, and the number of them is estimated to have fallen from 150,000 all the way to 25,000. They wanted you to find the percentage decrease in their number. So I'll find the difference in the numbers, meaning I'll find 150,000 minus 25,000. That is 125,000. And I'll divide this value by the higher value, which is 150,000. So again, I'll find the difference first, and then I'll divide by 150,000. And that is my final answer. 83% decrease in the number. That's a massive decrease. So it appears that this cancer the animal is suffering from is reducing their number. Okay? Now, next, they're telling me about the niche of this animal. They're telling me about the role of this animal in its habitat. So it feeds on dead bodies and it actually scares off other scavenger. What's a scavenger? A scavenger is like hyenas. They feed on dead animals. So in my table, I got before, that's before, and I got after, meaning when it was healthy and when it was sick. We have other scavengers here. They did not mention all scavengers in the habitat, they just mentioned 3. And you can see when it was strong, it was spending more time feeding on the carcass. Carcass, they told us about it. It's the corpse of dead animals. So when it was strong, it was like taking over the habitat. It was rolling over, it was scaring off all of the other scavengers. But when it's getting sick, it's spending less time. On the other hand, the other scavengers are now taking over. They're spending more time feeding. So that's my first piece. The second part, proportion of dead animals left on the ground. So again, this animal, when it was strong, we had less and less of dead animals left on the ground. It was eaten faster. But when it got sick, those dead animals are taking longer to disappear, to decompose. Okay? So, in brief, here, what is happening? We had a strong animal, it was scaring off all the other species in its habitat. Due to its sickness, it's getting weaker, it's spending less time feeding. Probably they're dying, a lot of them. And who's taking over the niche? Who's competing and taking over? It's actually the other scavengers, like the raven, the spotted-tailed, the feral cats. Okay, so the question is worth 6 marks, and they said explain the results of the study. Again, with a 6 mark question like this one, and it starts with explain, there is always a way to score 1 or even 2 marks by just describing that data. So I'll start like this. I'll say that animals spending less time, reason, because the question says explain, because it's dying and it's getting weaker. Okay, there's a fewer of them due to their death. What about the other scavengers? Those ones I have in the other columns here. Those are now spending more time. Okay. Reason, there's nobody to scare them off. Okay. And there is more food. How come there's more food now? Because the main scavenger was or is not competing anymore. So with the cancer, what is happening? It's becoming weaker. It's less likely to scare them off. Other animals are now present in similar proportions in both. But I'm talking now about animals that they did not mention. Why? How can I have animals that are not going to get affected? I may have animals that are not affected because they don't compete. You know, like when you have animals and competition, this is the niche of the first animal. This is the niche of the second animal. If two animals, A and B, and those two animals don't have overlapping niches, then if this animal A is decreasing in number, animal B is not going to get affected. But for my case, with the animals they mentioned here, they're all having overlapping niches. That's why those animals here are benefiting. Okay? So I need to mention the other case where there is no overlapping of their niches. That's what I stated here. Okay? So I'll say here that there's no competition in this case. Now, let's talk about the graph. With the case of the graph, you can see that there's more time those dead bodies are spending on the ground. But nonetheless, take a look at the bottom. Either way, those there's like complete decomposition. They're disappearing because decomposition doesn't rely on scavengers. Decomposition relies on decomposers, bacteria and fungi. So I said that here, say it's taken longer, but nonetheless, decomposers will, end of the day, they'll decompose the dead body. Okay? We're now getting to the last bit of this question. Explain what other biotic factors can affect the results. So they're saying that this experiment is not well-controlled experiment. There are other biotic factors that may affect it. Biotic means related to living things. So there could be other animals that they did not mention in this study. There could be humans, like humans hunt. Humans may scare off other animals. There could be disease, infections, bacteria, right? So I say here, it could be due to microorganisms. It could be due to scavengers that you did not mention. Could be due to humans. Could be to predators. What's the difference between like other scavengers and predators? Predators like carnivores, they're feeding on these scavengers. Finally, it's infections. Okay, and again, the question says explain. This means I need to write the factor and explain its role. Okay? We're now heading to question 5. Here they're asking me about a viral infection. When do we consider a viral infection? I may inhale a virus and I don't get an infection. A viral infection you will only get if the virus gets inside someone's cells, right? So it's a virus getting inside cells. The graph right here is about interferons. Like interferons are these proteins that help our cells fight viral infections. So here I have like a cell on the left. It got infected with a virus. This is part of non-specific immune responses like phagocytosis, interferons, inflammation. These are all non-specific. With this one is more related to viral infection, exclusive for viral infection. So if this cell got infected, it would signal with a protein known as interferons to the other cells nearby so that they prepare to stop the spread of viruses. How? Stopping their attachment, inhibiting its enzymes, or even trigger immune response, trigger a specific immune response. How? So that you see here, the cell is degrading the viral genome. That's one thing. And as I said, it may even stop the attachment of the virus or trigger phagocytosis of those viruses on the outside. So the question gave me graph, and it's saying here, explain the changes you have from day 0 all the way to day 7. So you can see here that the level of interferon was nearly 0. It was 0 here. Why it was 0? Because it takes time to synthesize the interferon. So even here, day 1, there was very limited amount of, then it increased, but then it decreased. What made that decrease? That's when the virus has been dealt with. The virus was phagocytosed. The virus was destroyed, so it's no longer a concern. So first of all, why it takes time? It takes time for the interferon to be synthesized. It's a protein. Protein synthesis takes time. Initially, there was no interferon because the viral infection did not happen. That's why they asked us this question at the beginning. So they want us to remember that the viral infection only occurs if the virus is inside the cell. Interferon production increases as the cells got infected, but then it falls when the virus has been destroyed. How can you destroy a virus? Non-specifically, by means of phagocytosis. Then they're asking me about specific and non-specific immune responses. So let's start with phagocytosis. Phagocytosis is non-specific, but it does also occur for specifics. Again, phagocytosis, if it happens as it is for some virus randomly is being engulfed, that's non-specific. But when the antigen is presented, then it will be essential for specific too. Both have the same purpose. Specific, non-specific immune responses have the exact same purpose. And presentation of an antigen is only part of a specific immune response. Okay? Next, they're asking me for a comparison between T killer versus T helper cells. So T killer is the one that responds to T helper, right? How does it respond? It receives a signal, cytokine, from T helper. Okay. So that is the way T killer works. T killer does what? T killer cells produce perforin, which is responsible to destroy cells that have been infected with viruses. So distinguish meaning only, I'm going to write differences. First one, T killer cells activate, sorry, T helper activate T killers. T killer cells destroy cells that have been infected with the virus. T helper produces cytokines. T killers produce perforin. T helper cells involved in what we call it humoral immune response, meaning it may activate also B cells, right? And B cells produce antibodies. But T killer is exclusive for cellular immunity. Okay? We have still 3 more questions to go. Question 6 talks still about immunity, I believe. Yeah, talks about immunity. It's talking about Alzheimer's disease, something not part of your syllabus, but they gave us enough information to understand how it works. They said if somebody has Alzheimer, then they'll deposit plaques where inside the brain. So with age, they deposit in their brain, they deposit some protein. This protein is called amyloid, and that causes what? That causes this disease. They found or they designed a drug. This drug is containing antibodies. And those antibodies do what? They target the protein responsible for Alzheimer's. So again, the question is like this. They're saying in somebody's brain who has Alzheimer, they deposit some protein. That protein, as it builds up, causes all the signs of Alzheimer, meaning they start to forget, etc. So their new drug is basically antibodies designed to be complementary to that protein. But antibodies, as they are, they're not going to destroy the protein. They're not going to remove the protein. This will be, do you remember that word when an antibody tags an antigen to be destroyed? This antibody will opsonize those proteins and then phagocytosis will clear up that protein, will remove that protein, and the person turns healthy again. Anyways, so the question says, describe information has been collected scientists before this drug could be approved. Do you know where this question is taken? It's taken from Unit 2, the part where it talks about the part that it talks about drug trials. So do you know what are the essential things to make a drug? First and most important, to ensure that the drug has minimum side effects. Second is efficacy. That's the phase 2. Then in phase 3, you design that perfect dose. And we may even consider the best way because this drug is targeting the brain. So we may even consider how are we going to deliver the drug? Is it going to be like pills, injections? And we may even see how long this drug has to be taken. Okay? So this is mostly Unit 2, the 3-phase drug trials. The question continues here. They said, explain the phrase antibody specific. So what does it mean when I say enzyme specific to substrate? That's similar, right? Complementary. Only binds to one substrate. Here's the same thing. Antibodies can only bind to specific antigen because they have complementary shape. Okay? Here it says, suggest how the drug could result in reduction in the amount of protein building up in the brain. So the story goes like this. The antibody binds to the antigen. Macrophages will now engulf because it's opsonization. That's when the target has been tagged to be destroyed by those macrophages. And do you know what macrophages do to break down that protein? The macrophages, as it engulfs, will destroy. It will break it down because it has that enzyme inside their lysosomes. They have hydrolytic enzymes inside their lysosomes. Okay? So just why a person did not have Alzheimer, why if we're getting those antibodies, we're extracting those antibodies to help someone else, why should we take antibody from someone who has never had Alzheimer? Simply because if somebody had Alzheimer, this means he couldn't make those antibodies in the first place. So again, I can't take antibodies from someone who struggles with Alzheimer because this means he failed to make those antibodies in the first place. For that reason, I would rather call for somebody who has already had B cells, had those memory B cells to make those antibodies so he can make those specific antibodies. Okay? Here comes the last bit of this question. Explain why the drug could be produced only from B memory cells taken from person who had the disease. That's what I was trying to tell you. It means that he has not have that in fact disease. He has memory cells. And do you remember that cell that makes like the actual cell that makes antibodies? It's the activated B cell. When a B cell has been activated, that's when it becomes the plasma cell. We have two more questions to go. My next question is about viruses. It's about Ebola. They gave me a diagram, quite useful because they show me that this Ebola here has polymerase. It has proteins in its capsid and has glycoprotein. So let's discuss first the RNA polymerase. Why could a virus have an enzyme? Viruses would have an enzyme if the host doesn't have this enzyme. So RNA polymerase here is essential because the viral genome for Ebola is just like HIV. In both cases, it's an RNA virus. Okay? So here, the RNA polymerase will copy the viral genome. What about those capsid proteins? The RNA will have the genetic code to synthesize these proteins. What about glycoproteins? Again, in the case of HIV, we have a glycoprotein known as GP120. Usually, when you have a glycoprotein on the surface of a virus, this glycoprotein is essential to unlock the cell, to trigger endocytosis in the host cell. Anyways, what does genome mean? The word genome stands for the genetic material. That codes for what? That codes for the essential proteins. And just state examples because it's worth 2 marks. I'll state examples of whatever proteins I have in the diagram. Okay? Next, they said, why does the virus require glycoprotein? That is essential because the virus needs to unlock host cells. So those glycoproteins, like here in my HIV, that's HIV here. With HIV, it binds with its GP120 to the host cell glycoprotein and it uses that to unlock the host cell. So I say attach first, specifically, and then it would hide inside the host cell. When it hides there, it becomes almost impossible to be destroyed by the B cells, the immune system. Why do we have RNA polymerase in such a virus? This is the case of RNA viruses like Ebola. There's no DNA here, so there's no DNA in such a virus. It goes RNA to protein, and that will be essential to make those proteins of the virus, the capsid protein. Now, just quick note here, the virus in this case is not relying on the host. The virus using its own enzyme. This means the host cell RNA polymerase is not functional in this case to replicate the viral RNA. So I'll say here to make copies of RNA plus to make messenger, which is reason because the host doesn't have such an enzyme. Like with the case of HIV, we have integrase, we have the reverse transcriptase. Those are not available in the host, so that's why the virus has them in itself. What about assembly? When I say viral assembly, that's when we pack the virus. We put the genome, the capsid, the enzymes all in one package. So I'll say this one here. That's when I assemble the RNA genome, the proteins, form the capsid, and we make new viral particles. Okay? Now, here comes the last bit of this question. It says, why structure Ebola includes VP35? They said that this VP35 inhibits interferons. Remember the interferon thing I was telling you about? That's the protein that moves from one cell to another, ensuring that the virus is not going to attach to the next cell. So just state how this will stop the action of interference. So say first, with interference is non-specific immune response is going to inhibit viral replication. It would also stop attachment, spreading of virus. So it increases the chance of the virus staying on the outside. If the virus is here, that becomes a very easy target for your B cells or the macrophages. Okay? My last question of the day talks about PCR, gene technology. It's about some birds here. They said that these crows look similar and they're difficult to be distinguished. As you can see, morphology here doesn't work. Just like because they're so similar. Give two differences between the structure of nuclear DNA and mitochondrial DNA. We took this quite early in Unit 2 that mitochondrial DNA is like bacterial DNA, like prokaryotic DNA is circular. Okay, so that's one easy difference. One is linear, one is circular. Okay. Second, it's histons. You know, like the histon thing is exclusive for eukaryotes. Mitochondria has prokaryotic origin. So you don't expect to have histons in the mitochondrial DNA. Moreover, I'm going to state two more things. They being a circle means what? Being circle means that nuclear DNA has overhanging phosphate at the end. Do you know, like every DNA strand has two endings, one end with a sugar and one end with phosphate, because the endings are free. The endings are not closed in a circle. Okay? But when you have circular DNA, this means what? This means I am ending up with some extra phosphodiester bond. Do you know what I mean here? I mean, if this is the DNA strand has phosphate, sugar in its backbone, right? Now, if this is linear DNA like the ones you have in your nucleus, then there's always a phosphate, there's always a deoxyribose at the end. They're free. However, if this was circular, then you'll end up with a phosphodiester bond right here. And that means what that means, I have one extra phosphodiester bond in circular DNA. On the next page, they gave me like the whole chart of PCR, like this. And they wanted me to do what? They wanted me to talk about three molecules other than nucleotides that would be needed to make, like to magnify DNA, to do PCR. I'll state primers. I need the building blocks to make DNA. I need the enzyme. Remember the name of the enzyme for PCR? We call it Taq polymerase. And I need optional, you may say, like buffer solution. So again, this said other than the sample DNA, where else do you need to magnify it? We got some easy math question here. You see that in the diagram, they gave me the time for each stage. Minutes, seconds, seconds, minutes, and they wanted you to convert all these into hours. So what am I going to do here? I'm going to take those values, turn them all to maybe minutes, and then see how many hours this will take. So I'm just going to do it like this. So all these values here in minutes, okay? Divide by 60 because they wanted the final answer to be in hours. Okay? 2 decimal points. So 1.38. And the last thing for today is this part. Explain how the process amplifies the using information in the diagram support your answer. This is when you need to have good memory about the temperatures and what each temperature is needed for. So to start with, the initial high temperature is needed to unzip DNA. We don't have in PCR, you don't have helicase. So we rely on high temperature to denature DNA. Then the lowest temperature is to bind the primers. 70 degrees is the temperature optimum for the enzyme. And then I have the role of the enzyme to align the nucleotides. Why did we do that 35 times? Look at the diagram. They said, you're going to do this 35 times, meaning you're going to have enough amount of DNA. Every cycle of PCR doubles the amount of DNA. Now, how do we see if there is any relationship between two species? To do so, I need to do what we call DNA profile. What's a DNA profile? That's when you run the DNA fragments on gel electrophoresis. If the species are related, then we compare them, compare those bands. Now, the more similar base sequences, the more related those species of birds are. Okay? Here's the last sentence. So this means we're done with this week's revision. Let me tell you the plan for the coming weeks. Like every week we'll meet this time, we'll do a revision, but at the same time, you'll be doing full past papers. We're going to start this week with 2019 past papers, and every week we'll be doing one full exam session. For now, we're going to start the test. Okay, I'm going to post your test on Google Classroom. So if you could just log into Google Classroom and start the test.